same time nanti Dr. Fauzin akan buat live. So tak pastilah live melalui Zoom ke Google Meet ke. Based on kuliah SOP, lab can be conducted if the number of students are less than 5 students. Okay, kita ada 3 orang yang ada dekat kampus. So mungkin boleh lah, tapi tak that sessions practicals kami perlu mohon kepada HOD Dr. Zarina. Okay, if Dr. Zarina okay, she's agree, next practical we will have panggil apa, hybrid mode, face to face dan juga online. Okay. So today we'll continue our discussions on growth and development. I'm not sure whether you have learned about embryogenesis. Ada belajar embryogenesis? Masa awak SPM, foundations ataupun dekat matric, tak pasti? Awak tak pasti ya? Saya pun tak pasti sebab saya dah tinggalkan SPM tu dah lama. Saya tak ingat saya ambil SPM tahun bila. Okay, so but during my undergrad, saya rasa undergrad saya tak ada kaitan dengan plant science pun. Maybe dekat foundation saya ada belajar, kan? So okay, tak apa kita because plant physiology will discuss basics of foundations knowledge on plants. So later untuk kelas-kelas macam genetics, seeds ataupun plant nutrition, you have a basic knowledge about that subject that you have learned in plant physiology. So for today we will discuss 3 topics which are embryogenesis, meristems and cell differentiations, plant growth and senescence. Okay, if you refer to our course outline, there are more topics should be discussed in growth and development. However, for example, cell walls, okay, kita dah discuss masa plant cells, but itu sebagai repetition. So kalau kita ikutkan betul-betul untuk course outline tu terlampau banyak yang perlu kita cover hari ni. Okay, tapi apa yang topic that we discuss today will cover about plant growth and also development. Okay, so what is embryogenesis? So basically from its words, embryogenesis refer to the development of embryo. So in plant, embryo can be developed from asexual or sexual reproduction process. Okay, for sexual reproduction process, embryo, the first stage of embryogenesis is the fusions of gametes and also sperms. And plant also is very unique. We can induce embryogenesis without sexual reproductions. Okay, awak belajar tissue culture, we can produce progeny plant seedling from vegetative cells of the plants. Embryogenesis may occur naturally in plant as a result of sexual fertilizations, the fusions of gametes, sperms and also ovums. And these embryos are called zygotic embryos and develops into seeds, which can germinate and give rise to seedling. So in sexual embryogenesis, the process will take place in an ovules, okay, a specialized structure forms within couples of flower. Okay, generally plant can be categorized into dicots or eudicots and also monocots. What's the difference between dicots and monocots? The cotyledons produced by the seeds during germinations. Okay, dicots will produce only one cotyledons, which dicots will produce 2 cotyledons. cotyledons. The dicot also at the initial stage, the dicots than monocots. But after globular states, the developments will be not slightly different, significantly different. Okay, later we will discuss the difference between embryogenesis in monocot and also dicots. So generally, embryogenesis in eudicots or dicots involve 5 different stages, which are zygotic stage, globular stage, heart stage, torpedo stage, and also major stage. Globular, heart, torpedo, the names derive from the shapes of the zygotes. Okay, zygotic stage is the first stage of embryogenesis in dicots and also monocots. Zygotic stage is the first stage of the diprot life cycles. The sperms and ovums contains haploids, half of genetic materials in plants, only one ends. After the fusions of sperms and also ovums, it will form zygotes. Okay, so this is zygote. The first one is zygotes, and then these zygotes will be differentiated growth, polarized growth, and produce asymmetric transverse divisions and giving rise to apical cells and also basal cells. Okay, later the apical cells will form tubular, whereas the basals will produce suspension. Okay, so kalau macam dekat haiwan macam kita, suspension ni macam umbilical cord. It will fuse to dekat biji benih, lupa lah nama dia, banyak benda sangat yang sih. Endosperms. Okay, it will fuse the suspension will extend, will growth, and the growth will not diffuse. The growth dia akan sampai ke endosperms, and then the functions of suspension later. Sebagai laluan untuk zygotes. Okay, and then the second stage is globular stage. What is globular stage? Globular stage is after 30 hours of fertilizations. Okay, so the apical, di sebelah atas kita panggil apical cells, will differentiate and produce 8 globular embryo cells. Okay, additional precise cells divisions increase the number of cells, okay, in the spheres. Okay, kalau kita tengok dekat sini, so this is basal cells. The upper part is apical cells. So the apical cells will divide into octant cells. Okay, kalau awak nampak dekat sini ada 4, 1, 2, 3, 4. Tapi kalau this is in 2D picture. Kalau dalam bentuk 3D, awak boleh bayangkan bola tu. So dia ada 1, 2, 3, 4. Belakang dia pun ada 4 lagi. So there are 8 cells, okay, in total in the apical embryo regions. Whereas the basal cells will produce one structure we call hypophysis. So hypophysis is the only structure from basal cells that will produce embryo. And then the outermost one layer thick cells, the next stage in globular stage, it will divide and produce one layer thick cells. These cells are called protoderms. Okay, proto, what is meant by proto? Apa dia proto? Selain protoderms, apa lagi dalam sains proto? Proto maksud dia maksudnya initials, okay. Later it will kalau protoderms, it will produce dermis, okay. So proto apa maksud dia? The initial stage. The next stage of layer cells from the protoderms will become the ground tissue meristems, and eventually it will form endodermis and also cortex. Okay, so this is the initial stage of globula. The apical regions, bahagian apical ni dia akan membahagi, okay, daripada 1 dia akan jadi 2, okay, lepas tu 2 kepada 8, okay. So octans, it will the number of cells, okay, will be n kuasa 2, okay. And then from globula, tadi sebelah luar, okay, for globula the outer layer will become protoderms, and then the next layer behind the protoderms will becomes meristem tissues, okay. And then the inner regions in globula will be produce procambiums, okay. And these procambiums later will become vesicle tissue and pericycle tissue in roots, okay. So this is peritoderms. The next layer is apa tadi? Meristems, and the last one will become vesicle tissues, okay. The apical regions, the apical regions give rise to the cotyledons and also shoots apical meristems. So apical regions adalah yang sebelah atas ni, okay. So it will develop and become heart shapes. The middle regions give rise to hypothetical roots and most of root meristems, and hypophyse give rise to the rest of the root meristems. Hypophyse is the only regions from basal cells that will contribute to zygote development. Heart stage, okay, the third stage, heart stage. Why it's named, the nama dia heart stage? Sebab dia punya bentuk macam heart ni, okay, kalau nampak. So ini nanti akan jadi cotyledons. So dia akan jadi dicotyledons, dia akan jadi 2 dicots. So this stage from through rapid cells divisions of 2 regions on each side of the embryo. So these 2 regions produce outgrowth that later will give rise to cotyledons and give the embryo bilateral stage, okay. The heart state, the center of this outgrowth will become the shoot apical meristem. So at this stage, the bilateral symmetry is established. Okay, bila the apical regions, the outgrowth, it will become like heart. Stems, okay, and then the hypophyse will become the root regions, okay, the root. Torpedo stage. This one is torpedo stage. Kenapa nama dia torpedo? Saya tengok bentuk dia pun tak torpedo, okay. Tapi if we observe under electron microscopes, some of the embryogenesis of monocotyledons, the shape will resembles like torpedo, okay. So this stage form as a result of the cell's elongations throughout the embryoaxis and also the further developments of the cotyledons. So the axis, the initial stage, the cotyledons will outgrowth and the shapes resemble heart shape. When it continues to elongate, the shapes will resemble torpedo. So sebab itulah stage torpedo. So toward the ends of embryogenesis, the embryo and seeds will lose water and become metabolically inactive as they enter dormancy. So ini bukan torpedo stage, sorry. This is a mature stage. Okay. So this is the mature stage of seeds. Bila dah sampai sini, so this stage is mature. It will lose water and enter dormancy periods. Okay. So depend on what kind of seeds. Certain seeds, their dormancy takes to several months, like oil palms, pokok kelapa sawit. The seed can last in dormancy periods for years. Okay, kalau awak ambil buah biji benih pokok sawit yang warna hitam, so awak try semai dia, it will take few months to germinate. So different seeds have different kita panggil apa? Different treatments to increase the germination rate. For example, for oil palms, macam ni kita nak increase nak break the seed dormancy by the heat treatments. We expose the seeds dekat 60 degree Celsius. If we expose at this temperature, biasanya dia punya protein akan degenerate and the seeds will become tak boleh hidup lah. But for pokok sawit, dia punya dormancy periods can be break by heat treatments. Okay. Sama juga macam kalau awak tengok berita dekat luar negara, especially dekat dekat Europe, kadang-kadang dia akan ada cuaca panas and then hutan dia terbakar. Lepas hutan dia terbakar, there are seeds that grow. Okay, so there are certain seeds that require very high temperature to break their dormancy. Okay, so ini stage kelima, bukan torpedo stage but mature stage. So this is SEM image of Arabidopsis thaliana. Okay, so if you kalau awak baca buku physiology, many of plant model used to study their genome sequence, their physiology, biasanya pokok Arabidopsis. Pokok Arabidopsis ni adalah pokok rumput. Okay, this is the initial stage. After the fusions of sperms and ovums, it will produce to regions, apical cells and also basal cells. Okay, so apical cells will contribute to embryogenesis. It will divide. Okay, becomes, this is the initial stage of globula. Dia ada 8. Tak berapa jelas Kalau awak nampak dekat atas ni ada 4, bahagian bawah dia ada 4. Okay, so this is the initial stage of globula, and then itu protoderms bahagian luar dia, and then it will further grow at their apicals, akan membentuk cotyledon, dia akan membentuk heart shapes, torpedo shapes, and then this one is mature stage. Okay, kalau awak nampak dekat yang sebelah sini, this is a seed coat. Okay, and then this one is the seeds and also the cotyledon. So that's the embryogenesis for dicots. For monocots, the process of embryogenesis quite complex, and it involves 6 different stages, which are zygotic stage, globular stage. So zygotic stage and globular stage in monocots are exactly same like in dicots. In zygotic stage, it will be initiated by the fusions of sperms and also ovums and produce diploid cells. Okay, these diploid cells will further divided to produce apical cells and also basal cells, okay, in a globular stage. And then they have transition stage, okay. So saya tak terangkan yang ni, sama saja. They have transition stage. What is transition stage? During the early transition stage, the scutellums appear on the in the inner side of the embryo. Okay, what is scutellums? So scutellums is organs. Kalau scutellums in insects, it refer to a protective layer of the insects. For seeds, scutellums um, scutellum is the single cotyledon of the grass embryo. The scutellum is specialized for nutrient absorptions from the endosperms. Okay, so tadi kalau dekat um, dicots, we have suspenser. Suspenser will further develops into endosperms. In embryogenesis, the 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 structure that will develops and pass through the nutrients from endosperms to embryo is scutellums. So ini adalah scutellums. Okay, so scutellums apart from and nutrients absorptions from endosperms to later embryo, it also serves as a protective layer, okay, to protect coleoptiles, okay, which protective layer for leaf, for nanti dia akan develop leaf, and also radicals, dia akan menghasilkan akar. So coleoptiles stage. This stage is marked by the formations of a distinct coleoptiles, scutellum, shoot apical meristems, root apical meristems, radicals, and also coleorhiza, protective sheets that cover the embryonic roots. Okay. So what is coleoptiles? Coleoptile is protective sheets. This one is coleoptiles. Okay. Ini adalah seeds of apa ya? Saya lupa lah pula, bukan padi, apa? Gandums. Okay, wheat. Protective sheets. Coleoptile is protective sheets cover the emerging shoots in monocotyledon such as grasses, in which few leaf primordia and shoots apex of monocot embryo remains enclosed. So the coleoptiles protects the first leaf as well as growing stems in seedling and eventually allows the first leaf to emerge. So if we compare to monocot, the monocot's embryogenesis will not develop this structure, coleoptiles. Okay. Leaf primordia stage. So the shoot's apical meristems initiate several leaves inside the coleoptile. Primordiums in embryology is an organs or tissue in its earlier recognizable stage of development. Okay, so this is coleoptile. And then under the coleoptiles, it will it has the structure apical meristems. Okay. So this one will produce leaf later when the seed germinates into a seedling. Coleorhiza will serve as a protective layer for roots. Okay, rhiza mean roots. Okay. And then the last one's maturation stage. During the final stage of embryogenesis, the affections of maturation sedated genes precedes the onsets of dormancy. Okay. Boleh kita rest 5 minutes kejap tak?
Boleh, Doktor.
Okay, Doktor.
Boleh.
Okay. So tadi saya sangat kelam kabut lah. Saya lepas ambil anak saya bawa dia untuk test. Tak sempat nak lunch. Saya nak minum air kejap. Dekat saya sakit lah. Sekejap ya. Okay, students, can we continue? So the embryogenesis in plants are differ from monocots and dicots. Okay, so what is the difference between monocot and dicots? Monocot are more complex compared to dicots. It involves 6 different stages. Okay, initial stages of embryogenesis in dicots and monocots are resemble sama. Okay, bila dia dah sampai globula in dicots they have globular stage, whereas uh, in monocots they have transition stage, colloidal stage, and also mature stage. So basically, embryogenesis established 2 basic developmental patterns that persist and can easily be seen in the adult plants. Okay, the apical basal axial developments or axial patternings, and also the radial patterns of tissues, for instance, and roots radial patternings. What is mean axial patternings and radial patternings? Bahasa mudahnya untuk kita faham apa dia? Kalau if you can explain in simplest word, apa dia? What is axial patternings? What is radial patternings? Okay, so uh, plant is different from animals and also humans. We humans and animals, our growth can be stopped at the certain stage. Okay, but for plant, their growth they can grow continuously. Okay, so during the embryogenesis, we can observe the patterns, axial patternings and also radial patterning. What is mean axial patterning? Axis, the growth excessive taxi. Okay, so if like us humans, our growth can be cannot be continuous. We will stop growth at a certain stage. Kalau macam lelaki, uh, biasanya umur 18 tahun. Kalau perempuan dia lebih cepat, umur 15 tahun. So our axial growth, okay, is limited. We will grow axially until a certain stage. After that, kita boleh radially, okay, kita boleh continuous growth lah, kan? But for plants, their axial, the growth can be continuous, axially and also radially. So this axial and radial growth, the patterns can be observed at the very early stage. At the globular stage, we can observe the differentiations of cells into a tissue that specialized later into um, kita panggil apa? Dermis, epidermis, cortex. Okay. So axial patterning established the apical basal axis. Okay. The shoot apical meristem is at one end, and the axis, the roots apical meristem at the other ends. Become evidence after the cotyledons. The first leaf begin to form at the ends of globular stage. So this growth, dia punya growth ni macam mana nak cakap, if we produce vegetative seedling from the plants, okay, for example, there are certain plant that can be grown vegetatively, macam pokok sugar canes. If you plant it in opposite directions, okay, yang bahagian atas tu waktu balikkan bahagian bawah, that part will grow as a, dia tak akan jadi akar, dia akan tumbuh tapi dia still bahagian pucuk dia akan terus differentiate the tissue, dia akan tumbuh sebagai pucuk dan akar dia akan tumbuh sebagai akar. Awak pernah buat tak? Pernah tak awak buat? Senyap je semua.
Tak.
Ngantuk ke? Awak tak pernah buat? Okay. So itulah kita cakap axial patternings. Okay. Kita nak panggil apa? Susun atur. apa ni kita panggil bahasa Inggeris susunan tu the alignments of tissues. Okay, for example, this is the axial patternings. Okay, so if we can observe these axial patternings start to develop, can be observed during the globula. Okay, this is the globula. The outer layer of this embryo will become epidermis. Okay, the inner one will become vascular tissues. Okay, so ini ada patternings. So because plant can grow continuously, so bahagian ni dia akan differentiate into dead tissues. Okay, kalau pokok tu hidup 100 tahun, dia akan hidup. Okay, tak seperti manusia atau haiwan. If we injured, for example, okay, hilang sebagian anggota badan, we cannot produce that organs. But for plant, if that organs become detached from the pokok ataupun become injured, these cells can grow and produce sel yang sama. Okay, radial patternings. Okay, so different tissue are organized in a precise patterns within plant organs. Okay, so radial patternings produce 3 tissue systems. The outermost layer of the epidermis tissue cover a cylinder of cortical tissue, which in turn overlies the vascular cylinder. Okay, so kalau we make cross sections at stems, we will observe these arrangements at the stems. Okay, and then if we make a cross sections at the basal stem, okay, apical stem, basal stems, we will observe the same arrangements. Okay, so these arrangements are initial or start during embryogenesis, and it will continue during the plant life cycles. Okay, so any questions? Faham tak? Tengah hari ni macam penat sikit kan, semua senyap je. Sebab selalunya waktu ni awak buat apa? Awak buat apa selalunya? Ya?
Just nak confirmation je lah yang antara axial dengan radial patterning tu, maksudnya kalau axial tu dia pokok tu makin meninggi kan, lepas tu radial tu pokok tu makin melebar, macam tu ke?
Yes, axials, okay, axials patternings. So axials patternings, macam saya cakap tadi, plant can grow continuously, tak macam kita. And kita grow sampai certain stage, our growth will stop. But for plant, they will continuously grow. And axials patternings kita dah nampak daripada stage embryogenesis, which part will become roots, which part will become shoots. Okay, and at the same time itu axial patterning. At the same time, when we make a cross sections, kita akan nampak patternings of the cells arrangements. Okay, dekat luar sekali dia ada epidermis, lepas tu cortex, and then vascular tissues. Okay, so that patterns, kalau kita make a cross sections at the upper part stems ataupun kita buat dekat roots, we will observe the same patterns, the same arrangements. So this arrangement, mula-mula sekali bila sperms and ovums to fertilize, dia tak ada specialized cells. Okay, and then at the globula, they start to specialize, become patterns. Okay, bahagian mana yang akan jadi pucuk, bahagian mana jadi akar, bahagian mana yang untuk yang stems dia susun atur dia, mana epidermis, cortex, and so vascular tissues. Okay, so this is a seed. Okay, ini adalah seed sebenarnya, tapi gambar dia tak penuh. Sepatutnya dia akan jadi macam ni. Okay, so ini adalah bahagian ni, we without endosperms. Okay, we have endosperms. Lepas tu kita ada seed coat sebelah atas dia. Okay, so at the mature stage, semua seed dia akan jadi kering kan. Kalau nampak pokok pun, at the mature stage, the seed will lose water, become dormant. How to initiate germinations? By introduce water. So the seeds will absorb water. Okay, it will activate certain enzymes, and these enzymes it will activate certain hormones, gibberellic hormones, and it will start to germinate. Okay. So any questions? Any questions? So let us proceed with second topics, which is meristems and also cells differentiations. So what is meristematic tissues or meristematic cells? So meristem cells or meristem tissues are resembles to stem tissues in our body, humans. Okay, so these meristems and also stem tissues or stem cells is undifferentiated cells. So now scientists, for example dekat kuliah kita pun ada Dr. Hamzah dia buat how to induce. Okay, for example we have stem cells. How these cells can we induce their growth and differentiations and become certain tissues or organs. So if we can do that later, kita juga boleh macam jadi tumbuhan. Okay, for example, untuk sekarang dah ada orang buat. Okay, for example, they are very well known specialist doctor yang sekarang dekat New Zealand. So doktor tu asalnya adalah kelahiran Malaysia, tapi tak tahu kenapa dia lebih suka hidup dekat untuk berkhidmat di New Zealand. Okay, so for example, kalau certain orang boleh dapat kecacatan daripada kemalangan. Okay, for example, we can a person can suffer from melecur daripada kebakaran. Okay, there are different degrees of burning kan, melecur. Kalau degree yang sangat tinggi, teruk, dia akan jadi parut, dia tak boleh tumbuh semula. So nowadays, apart from doing plastic surgeries, we can treat that scars using stem cells. Okay, we can induce that cells to a specialized cells. Okay, for example, stem cells ni sel yang tak ada specialized lagi. We can induce the cells become epidermis. Okay, so dia akan menggantikan bahagian-bahagian yang rosak tu tadi. So to induce the cells to become tissues, yes, we can do that. Tapi kalau kita nak induce that cells to become to generate organs, setakat ni tak boleh lagi. Okay, but for plant we can do that. Kan, kita boleh buat tu. Sebagai contoh, kita ambil meristems plant, we can induce using gibberellic acids or auxins, and we can produce a new plants, okay, a seedlings, okay, melalui tissue culture, kan. So itu adalah background what is meristematic cells ataupun dekat humans or dekat animals stem cells. So for these cells, there are certain characteristics. Okay, the obvious ones, what is the characteristic of meristematic cells? It's not differentiates into specific cells. So meristematic cells are groups of undifferentiated cells that are in continuous cell divisions. So differentiations, what is differentiations? Differentiations is the process by which cells acquire metabolic structures and also functional properties that are distinct from those of its progenitor cells. Okay, for example, our stem cells, biasanya kalau dekat manusia kita punya stem cell dekat tulang sumsum. So these cells is not differentiate. Okay, they can continuously divide After it differentiate into specific tissues or organs, it will stop differentiate. So meristematic cells are typically small and nearly spherical. They have a dense cytoplasms and relatively few small vacuoles. Meristematic cells can retain its embryogenic characteristics. What is meant by embryogenic character? So meristematic tissues in plants can retain its embryogenic characteristics indefinitely. Okay. The reasons for this ability is that some meristematic cells do not become committed to differentiation pathway, and they retain the capacity of cell divisions as long as the meristematic remains vegetative. Okay, so this is stem cells, okay, or meristematic cells in plant. Okay, so these cells will differentiate. So the difference, the divisions rates, okay, of meristematic cells and stem cells compared to the specialized cells is different. Okay, biasanya stem cells and meristematic cells, their divisions rate sangat lambat. Okay, for example, in here, initially we have one meristematic cells and divided into two meristematic cells. From two, it then further divided into four. But for meristematic stems, usually there are certain daughter cells that remain as a meristematic cells, whereas the other daughter cells will specialize, committed to a certain pathway and differentiate into certain cells, differentiate into specialized cells. Okay, for example, it becomes vascular cells ataupun dia menghasilkan daun, dia menghasilkan bunga. Okay. When meristem cells divide, on average, one of the daughter cells will retain the identity of the stem cells. Okay, so macam yang saya terangkan tadi, meristem cells will divide. Okay, at the rate slower than differentiate cells. And when the meristematic cells differentiates, at least one of the daughter cells will retain kekal remain as a meristematic cells. Okay, so meristematic cells usually divide slow compared to other committed daughter cells. Okay. However, it may enter a period of rapid cells divisions before they stop dividing and can be recognized as specific cell types. Okay, so there are few types of meristematic cells. So why plant can grow continuously? Sebab apa? Why plant can continuously grow? Sebab apa?
ada meristematic
Ada
cell tu.
AZRI patterning.
Okay, so tadi, so in plant apicals or basal, it will contain meristematic cells. Okay, it will divide into shoots, or it will divide it into roots. When it divided into roots, at least one of the daughter cells will remain as meristem cells. What is meristem cells? Undifferentiated cells, unspecialized cells. Okay, dia tak jadi specific cell types, they remain as meristem cells. So that's why plant can continuously grow. Okay, because it can retain these meristematic cells. Okay, bila dia dah tak jadi meristematic cells ni, dia dah tak boleh membahagi lagi dengan dia tak boleh, bukan membahagi, differentiate into a new specialized cells. Okay, so there is few types of meristematic cells that or tissues that can be found in plants. Okay, we can find apical meristems. Apical meristems is meristematic tissues that located at root and also shoot tips. Okay, so that's why plant shoots and plant roots can continuously grow because it contains meristematic tissues. Okay, So apart from that, it also can it also contains lateral meristems or meristematic tissues. So this is the meristematic tissues located in the vascular and also port cambia. Okay, dekat lateral. So sebab itulah plant can grow axially and also radially. And in plant also it contains intercalary meristems. Okay, so these meristem tissues are located at internodes or stems regions between the place at which leaves attach and leaf bases, especially of certain monocotyledons. Okay, so for monocotyledons or plant that can kita propagate vegetatively, kita boleh potong kan, sebagai contoh tadi pokok sugar cane, pokok tebu, kita boleh potong and it can grow roots and shoot dekat internodes tu. Sebab apa? Dekat bahagian tu ada intercalary dekat internode tu it contains intercalary meristem. Okay, so what is the functions of apical meristems, lateral meristems, and also intercalary meristems? So apical meristems will give rise to the primary plant body. Okay, what is primary plant body? It will produce shoots. It will produce from shoot. It will mature, become stems, and it will produce roots. Lateral meristems is known as secondary meristems, or it involved in secondary growth because it increase in stem girth and also diameter. Okay, meristem form anew from other cells in injured tissue. Meristem forms anew cells, okay, from other cells in the injured tissues and are responsible for wound healings. So ayat dia tak betul, nanti saya type balik. Okay, meristem forms anew forms of cells, okay, in the injured tissues and responsible for wound healings. Plant continue to grow throughout their entire lifespan because of unlimited divisions of meristematic regions. Okay, so this meristematic part tu yang akan sentiasa membagi dan differentiate into new tissues or new organs. Meristematic self-differentiations, there are certain meristematic stems, meristematic tissues that can differentiate determinants, and there are also other meristematic tissues that undergoes indeterminate differentiations. What is means by determinates and indeterminates? Determinates genetically programs limits to their growth. Indeterminates showing no predetermined limit to growth condition as long as resource permitted. So sebagai contoh, determinates growth apa dia? Dekat leaf, fruits, flower. Okay, indeterminates dekat apicals, basals, dekat pucuk dan dekat roof. Okay, the vegetative shoots apical meristems usually is indeterminate. Okay, macam saya cakap tadi, shoots, roots, it usually indeterminate differentiations. They repetitively forms phytomers. Okay, kita ada monomer, isomer, photomers apa? Photomers adalah satu unit for kita panggil apa? Plant organs. Okay. For example, photomers are plant units that consist leaf, nodes, internodes, and axillary bulb. Okay. So dekat sini we have apical meristematic. So this apical meristematic tissues can differentiate indeterminately. Okay, it can produce flower, leaf, roots from here. Okay, and kalau kita nak buat tissue culture pun biasanya kita ambil daripada bahagian yang ini kan, determinate meristematic tissues. Okay, Berapa dah? 6.4. Okay, saya ingat saya boleh habiskan dalam sejam. Tak boleh ya. So ada few slides. Oh, ada lagi tak sampai 10 slide. Okay. Any questions? Any question?
No
Tak ada ya?
question.
No questions. Okay, we move to plant growth. Yeah.
Doktor, ada
Yes.
orang request nak masuk kelas.
Ada orang request. Siapa ni? Inshira. Hai Inshira, awak baru masuk ke tadi dah masuk terkeluar balik?
Baru masuk doktor.
Baru masuk ya, okay. It's okay.
Ah,
So, saya,
sorry,
it's okay.
terlupa doktor. Sorry.
Terlupa. Okay lah. So, saya pun hari ni macam tak bersemangat, sorry lah. Okay. Tadi saya dapat yang message yang ada positive cases dekat nursery anak saya. So hopefully anak saya tak ada positif lah. Saya tadi tak sempat nak beli RTK. Anak saya tak ada apa-apa. And then tu dah buat saya tak ada mood. Okay, risau pasal anak saya. And then tadi kakak saya pula call. Dia, her husbands and their 2 kids who test positive for COVID. So sebelum ni kita dah dekat 2 tahun COVID ya. Saya syukur sangatlah tak ada pun ahli keluarga saya, adik-beradik saya yang kena COVID. Okay, keluarga saya, keluarga kandung saya ataupun keluarga isteri saya. Tapi hari ni untuk rekod the first case dalam keluarga saya ada kes COVID ni. Okay, so hopefully lah saya doakan awak semua tak adalah yang kena jangkitan COVID ni. Okay, so can we continue to our last topic, which are plant growth and also senescence. Okay.
Yes.
So what is plant growth? What is plant growth? So for plant, dia punya growth definitions will be different. Okay, kalau kita animals, bacteria, the growth definitions will be different. For plant, irreversible increase in volumes, okay, because plant continuously growth, the volumes will continuously increase, and this increase are irreversible. Okay, the largest components of plant growth is cell expansions driven by turbo pressure. Okay? During this process, cell.
Doktor,
Yes.
sorry ganggu doktor tengah share screen ke? Tak, kita tak nampak.
Ya, tak nampak ke? Tak ada ya. Oh,
Tak ada.
sorry. Okay. Boleh nampak ya? So this is our last topics, growth and also senescence. Okay. So even though plant can grow continuously, enter a senescence stage. Okay. Apa itu senescence? Apa itu growth? Kita will discuss in these topics. So plant growth is defined as an irreversible increase in volumes. The largest components of plant growth is cell expansions driven by turbo pressure. Okay, so during this process, cell will increase its volumes, manifolds, and also becomes highly voculated. However, size is only criterions that may be used to measure growth. Okay, if we measure the volumes, we only measure the size. Sometimes plant can increase its growth without increasing its size. Okay, that's when their cells increase in number but not increase in volumes. Okay, so if you want to measure the growth, make sure you use a correct parameters. Okay, because like I said earlier, plant can increase the number of cells without increase its volumes. Okay, growth also can be measured in terms of change of fresh weight, fresh weight. So tadi kalau kita nak ukur plant berdasarkan size, okay, yes, it can give us some growth parameter of plants. Okay, for example, if you measure the height, the diameter, the girth of the plants, we can measure by measuring this parameter, we can estimate the plant growth. Okay, so in other words, if we measure the height, the diameter, the girth of the stems, we will measure the size. However, in certain conditions, plant can increase its growth without increasing its size. Macam saya cakap tadi, it will produce high number. The cells, the number of cells increase without increase its volumes. Okay, so in these conditions, kita tak dapat measure the plant growth accurately. Okay, for example, the volumes, the size also affected by the turgor, the apa kita panggil the turgor pressure of the plants. For example, if you measure herbaceous plants during in the mornings, okay, for example, its height, okay, when its turgor very high, kita nampak tinggi dia meningkat, okay. Tapi bila tengah hari tu, when a transpiration rate, the turgor pressure in plant reduce, kita nampak dia sedikit mengacut. Okay, so if we measure its size in certain conditions, it will not be really accurate. Okay, so how we can measure the growth accurately? Depend on the situations. Tak semestinya kalau kita measure diameter betul atau salah, depends pada kita punya situasi. Okay, growth also can be measured in terms of change in fresh weight. Okay? That is the weight of living tissues over the particular periods of times. Okay, for example, tadi bila kita measure volumes in certain conditions, it will not give a very accurate measurements. We can measure the plant growth in another ways by measuring its fresh weight. Okay, however, fresh weight are affected, okay, by the water status in plant. Sama macam volume tadi, atau size tadi. Kalau kalau kita ambil pokok tu waktu pagi, where the plant turgor very high, kita dapat fresh weight dia sedikit tinggi. Compared if we harvest the plant during afternoons, where the transpiration process are very high, the same plant will produce a different measurements, will give different weights. Okay, so in these situations, measurements of dry weight are often more appropriate. So selalunya kita tak akan ambil satu parameter saja to determine the plant growth. For example, if you do experiments, you apply a fertilizer and you want to determine whether the plant growth are affected with the applied fertilizer. So to determine the plant growth, usually we will measure difference of few parameters. Okay, for example, we take the diameter measurements, we take the height measurements. Apart from that, we also take fresh weight and also we take dry weights. Okay, so in that way, kita dapat determines the plant growth accurately. Okay, so when you, macam saya cakap tadi, when you take measurements on plant growth, you must consider a few factors. Okay, if you want to take the size, so size are affected by the trigger pressure. If you want to weight, you must make sure whether you want to measure the weight during fresh weight ataupun after awak dah dry kan, and then awak dapat dry weights. Okay, cells number. Okay, so poses problems in multicellular plants. Okay, so we are very difficult to measure the cells number in plants. Okay, so in certain organisms we can measure its growth by measuring or estimating or enumerate the cells number. For plants, it difficult for us, not only in plants, in other multicellular organisms, it's very difficult for us to estimate the cells number. Okay, for example, like I mentioned earlier, if we take root measurements or the diameter of the stems. Okay, so for example, stems, the volumes, the amount of cells dalam sebagai contoh 1 cm persegi dekat stems punya cells number will be different if we measure the number of cells in leaf, for example. Okay, faham tak? Kalau kita ambil dua organs tumbuhan, for example, we take leaf and also stem. Okay, so 1 cm per 1 cm cubics of stems will contain different amount number of cells compared to 1 cm cubics of leaf. Okay, if that organisms are unicells, for example bacteria, yes, we can use the number of cells as the growth parameters. Okay, kalau dekat bacteria biasanya kita akan measure the populations, cells number to represent the growth of that microorganisms. But for multicellular macam plants, macam haywans, kita tak boleh buat macam tu sebab different organs dia punya number of cells will be different. Okay, plant senescence. What is plant senescence? Even though plant can grow continuously at a certain stage, sometimes the whole plants or certain organs, it will enter senescence periods. What is senescence? In biology, senescence is a process by which cells ages and permanently stops dividing but does not die. Okay, so to recover a portions of the valuable resources that plants invested in leaf formations or organ formations. Okay, so why plants undergo senescence and also kita panggil apa selain lepas senescence, cell death. So not like animals or humans, plant can recycle its nutrients within its tissues. Okay, for example, if the plant want to use nitrogens, okay, it will take the nitrogens from senescence organs and translocate that nitrogens into newly formed organs. Okay, for example, dekat daun yang dah tua. So plant takes the chlorophylls in that leaf and break down that chlorophyll into elements, nitrogens, magnesiums, carbons, and translocate that elements to newly formed organs. Okay. The senescence in plant can caused by the internals, monocarpic senescence or externals. Okay, what is monocarpic senescence? Apa the monocarpic senescence? We have perennial plants and we have seasonal plants. So monocarpic perennials adalah when plants dia dekat life cycles dia dah become mature, it will undergo senescence. Okay, sebagai contoh pokok jagung. Pokok jagung, the life cycles from seeds, the seeds will germinate into seedlings, seedling become mature, and then it will produce seeds, and then senescence. Okay. For external senescence, sometimes it affected by the abiotic. Okay. Sebagai contoh, temperature. Okay, certain pokok bila masuk musim sejuk dia akan jadi lower, so dia akan enter senescence process. Okay. Few types of plant senescence. Seasonal leaf senescence. Okay, in the 4 seasons climates, the plants will undergo this seasonal leaf senescence. Sequential leaf senescence. Okay, sequential leaf senescence due to the age, when the plants become mature, and then it will enter senescence phase. Senescence of fleshy fruits. Okay, ini adalah ripenings. Okay, the mature, these organs will become mature. Okay, so senescence process will occur in an ordered series. Okay, for example, the first stage, the chloroplast will deteriorate. Okay, it will undergo degradations. The chlorophylls will kita panggil apa? The catabolic process will happens to chloroplast. It will degrade or deteriorates into its elements. Okay, macam chloroplast, dia akan degrade into nitrogens, magnesiums, and also carbons. Okay, so these elements can be translocated into new organs where the demands of these elements are high compared to the senescence part of the plants. Any questions? Any questions? Kenapa semua senyap saya? No questions. Okay, for quiz, saya akan upload malam ni. For quiz, okay, for second quiz. Tadi saya sempat check soalan yang sesuai. I will ask questions from topic that we discuss in minggu ketiga dengan minggu keempat. So minggu ni saya tak akan masukkan soalan untuk quiz. Okay. So for example, kalau minggu sebelum ni untuk quiz saya dah masukkan topik active transport or passive transport. So that topic will not be asked again in your midterms. Okay. So faham tak?
Faham. Okay.
Okay. So any questions? Any queries or any other topics that you need further explanations? Semua okay? Syuhada, okay Syuhada.
Doktor, yang plant senescence tu kan, kalau yang pisang tak berbuah 2 kali tu kira plant senescence ke?
So, macam saya cakap tadi, there are seasonal plants, there are perennial plants. Okay, what is seasonal plants? What is perennial plants? So, perennial plants are plants that can continuously grow. Okay, even though before this we discussed I mentioned that plant can continuously grow. This is true for perennial plants. Examples of perennial plants apa dia? Pokok sawit, pokok getah, pokok kelapa. So that's perennial plants. For seasonal plant, their life cycles their growth are limited. Their growth can be stopped at certain stage. For seasonal plant, their life cycle begins with the seed germinations, become seedlings, and seedling become mature plants, and they produce fruits and seeds, and it will enter the first stage tadi, dia akan germinate balik. Okay. Untuk pokok pisang dia jenis apa? Dia seasonal plant. Kan, dia ada anak pokok, dia hidup, dia jadi it grow to mature plants, produce fruits, and then it will die. Okay, so the life cycles dia macam tu. Okay, so kenapa orang Melayu cakap pisang tak berbuah 2 kali? Pisang kalau berbuah banyak kali kan bagus. Sebab apa? Pisang lepas dia buah apa jadi? Kenapa pisang berbuah 2 kali tu baguslah kan pisang berbuah 2 kali, tapi kalau macam perumpamaan orang Melayu tu, pisang berbuah 2 kali tu refer kepada benda yang kejadian yang tak elok. Kenapa ya? Why? Tak ada idea.
Tahulah.
So sebenarnya soalan tu saya pernah tanya dekat cikgu saya masa saya sekolah menengah dulu. Cakap masa tu belajar perumpamaan. So kalau pisang berbuah 2 kali tu it refer to kejadian yang tak elok kan. Kita tak nak pisang berbuah 2 kali. Tapi saya cakap bukan ke kalau pisang berbuah banyak kali bagus. So pisang bila dia berbuah apa yang jadi? Macam mana kita nak ambil pisang tu? Kita potong dia. Kita tebang dia kan. So pisang tu bila kita dah potong, tebang, dia tak akan tumbuh balik. So dia merajuk, cikgu saya cakap. So that refer to macam tu lah. Saya pun sampai sekarang macam tak sesuai je kita nak refer kejadian tak elok dengan pisang berbuah 2 kali. Okay, itu sajalah saya cerita pengalaman. Apa, any other questions? Ada apa-apa soalan lain? Okay, tak ada soalan ni. So next week Dr. Fauzan will take over this class, and then pada minggu ke-8 awak akan ada midterms. Okay, so midterms adalah soalan-soalan daripada saya punya topik, daripada topik seminggu pertama kepada sampailah topik seminggu ke-5. Okay, so thank you for your time to listen to our discussions today. Later, if you have any queries or questions, you are always welcome to give me a message or ataupun awak nak call saya, boleh ya. Okay, thank you. Assalamualaikum. Jumpa lagi next time.